In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s, but in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy.
There are as many molecular machines in the human body as there are functions that the body has to do. So if you think about hearing, seeing, smelling, tasting, feeling, blood clotting, respiratory action, the immune response, all of those require a host of machines.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
This is absolutely mind-boggling to perceive at this scale of size such a finely tuned apparatus, a device that bears the marks of intelligent design and manufacture. And we have the details of an immensely complex molecular realm of genetic information processing. And it's exactly this new realm of molecular genetics where we see the most compelling evidence of design on the Earth.